DNA, which is short for deoxyribonucleic acid, is a genetic material that makes up a gene. A gene is a unit of heredity that transfers genetic information from parents to offspring. This is a single strand of DNA in which genetic information is encoded into 4 types of nucleotides: A, T, G, and C. A nucleotide is a monomer that is composed of a nitrogenous base, a pentose or sugar, and 1 or more phosphate groups. These monomers are grouped into pyrimidines and purines. The pyrimidines consist of C, cytosine, T, thymine, and U, uracil. The purines consist of A, adenine, and G, guanine. In RNA, the nucleotide pairs are A to U and G to C, and vice versa. In DNA, it's A to T instead of A to U, and G to C is the same. RNA modification is the process of altering the amino acid sequence by modifying the RNA nucleotides. Most of these changes are called point mutations. The first is a missense mutation. This is when a single nucleotide changes and a different amino acid is produced. The second is a silent mutation. This is when a nucleotide changes, but the amino acid produced does not change. Last is the nonsense mutation, which is when the nucleotide changes and its corresponding codon turns into a stop codon, thus stopping a sequence. Finally, we have insertion and deletions. This is when a nucleotide is either added or removed, thus resulting in a frameshift mutation. mRNA, also known as messenger RNA, is an RNA molecule that carries genetic information from the DNA to the protein synthesizing part of the cell. RNA polymerase is an enzyme that pries the 2 strands of DNA apart and joins together RNA nucleotides from the DNA template strand. The poly A tail is on the 3' end of the strand and is a chain of 50 to 250 more adenine. The 5' is the 5' end and is a modified form of guanine. They both are used during the export of mRNA. They both protect the mRNA, and they both help ribosomes attach to the 5' end. Translation is the synthesis of a polypeptide using the information in the mRNA. You start with the nucleotide sequence of the mRNA, and you translate that into the amino acid sequence. There is a small and a large ribosomal subunit. The small one binds to a molecule of mRNA and reads it. The large one joins amino acids to form a polypeptide chain. In the large ribosomal subunit, there is the A, P, and E site. The P site holds the tRNA with the polypeptide chain, the A site holds the tRNA with the next amino acid, and the E site is where discharged tRNA exits. tRNA, also known as transfer RNA, is the RNA that moves the amino acid from the cytoplasm to the growing polypeptide chain. rRNA, also known as ribosomal RNA, is combined with proteins to form small and large ribosomal subunits. First, the RNA genes are transcribed inside the nucleus. Then the rRNA is processed and assembled with proteins from the cytoplasm. And last, the subunits are exported via nuclear pores to the cytoplasm. A start codon, usually methionine, is a specific codon that signals for the protein synthesizing machinery to start translating mRNA. Aminoacyl tRNA synthase is an enzyme that correctly matches up tRNA and its corresponding amino acid. First, the active site binds the amino acid in the ATP. Second, the ATP loses 2 P groups and binds to amino acid as AMP. Next, the correct tRNA covalently bonds to the amino acid replacing AMP. And last, the tRNA and the amino acid is released by the enzyme. An amino acid, or AA, is an organic molecule containing both an amino group and a carboxyl group. Peptidyltransferase is an enzyme that catalyzes the addition of amino acid residue in order to grow the polypeptide chain. Polypeptide is when one or more polymers of amino acids are bonded together to form a chain. This shows two amino acids bonding together to form a polypeptide. The codon is the nucleotide triplet of mRNA used to determine the amino acid being produced. Stop codons, or termination codons, are codons that do not designate an amino acid and they mark the end of translation. An anticodon is a tRNA nucleotide triplet that base pairs with its complementary codon on the mRNA. Initiation is the process that brings together mRNA, a tRNA with the first amino acid of the polypeptide, and two ribosomal subunits. First, a small ribosomal subunit binds to a molecule of mRNA. Then, a tRNA base pairs with the start codon. Second, the arrival of a large ribosomal subunit completes initiation. Elongation is the process where amino acids are added one by one to the previous amino acid chain. First, the anticodon of the incoming amino acid tRNA base pairs with the complementary mRNA. Next, an rRNA molecule of the large subunit catalyzes to form a bond between the A site amino acid and the P site amino acid. Last, the subunit moves the A site tRNA to P site, while empty P site tRNA is moved to E site and is released from the enzyme. Termination begins once a stop codon reaches A site. A release factor then binds to the stop codon and adds a water molecule, which breaks the bonds between the P site and the completed polypeptide. Introns, or intervening sequences, are segments of nucleic acid that go between coding regions. Exons are the other regions of an RNA/DNA molecule that are usually translated into amino acid sequences.